Okay, guys, welcome to topic 6. So, DNA is made of monomers. Now, a monomer is in DNA is known as a nucleotide, and that's made of the following: a phosphate combined to a pentose sugar and a base. So, all of this is a nucleotide. This DNA molecule, this over here, let's say one of these would be a nucleotide. So, all of that is equivalent to this. As you can see, there's many of them joined together. So DNA is basically a polymer, but a polynucleotide. If they ask you to explain the structure of DNA, you say the following. DNA is made of 2 strands, and it has a double helix structure. This structure here, that you can see, like a staircase, is known as a double helix structure. There are 4 bases. C binds to G, so if you have C on one side, on the other side you're going to have G, and A with T. So if you have A, on the other side you have T. Okay, so DNA is a polymer made of many nucleotides, and it has a double helix structure. Now, what's the function of DNA? Well, DNA contains genes. A gene is just a section of DNA, and genes are responsible for sequencing amino acids in the cell. So the order of bases in the gene determine the order of the sequence of amino acids. The sequence of amino acids then determine what protein is made, which kind of whether it's an enzyme, an antibody, etc. And then those proteins determine our characteristics. So make sure you remember this sequence. Now, what happens in a mutation? So first of all, a mutation is a change in the DNA-based sequence. How can a mutation affect our characteristics? So here's an example with how it can affect an enzyme. So if the DNA gets changed, because of a mutation, then the next thing that's going to get affected is the sequence of amino acids. They're going to change the order as well. If the sequence of amino acids change, that means the shape of our protein is going to change. And finally, if the shape changes, let's say, for example, in this enzyme, we know that the active site is very important for the shape. If that changes, then there's no active site, so the function of the protein will change. So that's an example of how mutations can affect our characteristics. Okay, reproduction. There are 2 categories. You have sexual reproduction and asexual. So there's pros and cons, or rather there's differences between them. Sexual involves 2 parents. Usually all animals have sexual reproduction. Asexual is 1 parent. So usually like fungi, they can replicate. And they will have asexual reproduction. So there's no fusion of gametes. Gametes means sperm and egg. Therefore, all the offspring are going to be identical to one another. So if you have genetically identical offspring or individuals, they are called clones. Okay, meiosis. Now, meiosis is we did mitosis in the first paper. Meiosis is the production of sperm and egg. So mitosis was for body cells. Meiosis is only for production of sperm and egg. And the way it works is the following. There are 2 divisions, first of all. So in mitosis, there was 1 division. Here, there are 2 divisions. So in meiosis, first of all, the chromosomes line up in pairs in the nucleus. Then what happens is that the pairs separate. So for example, this little guy will go one side, and the other one will go to the other side. Once they separate, then the nucleus, then the cell divides, and there you go, look. We have 2 cells, and you can see they're no longer in pairs, they're in single file. Again, now what's going to happen is the arms are going to separate. So the sister chromatids are going to separate, and again that's going to divide into 2 more cells, each of them. So in total you have 4 cells. Now how would you explain that in your exam? These are the points that you have to mention. Step 1, DNA replicates. Step 2, chromosomes align in pairs in the center of the cell, just like this. Step 3, the pairs get pulled apart. Step 4, in the second division, they line up again, but in single file. Now the arms get pulled apart. And finally, 4 daughter cells, which are all genetically different, and haploids are produced. A diploid is a cell that has 23 pairs of chromosomes. A haploid is a cell that has only got 23 chromosomes. Okay, moving on to genetics. So you have the words genotype and phenotype. Genotype just means what sequence of alleles you have, or what genes you have, whereas phenotype is what visible characteristics you have. So if we look at this example, we can see over here that, let's say, B and B, big B and small b, are the different alleles for the gene for hair color. So we can see that big B is for brown hair and little b is for blonde hair. Also, big letters are dominant small letters are recessive. So, how would you describe this particular individual? Well, because he's got two big B's, we would say that it is homozygous. Oh, sorry. Homozygous dominant. And this person over here is also homozygous because they have the same letters, but they are homozygous recessive because they have two small letters. So on the left you have homozygous dominant, on the right you have homozygous recessive. And the person in the middle, this one, has a big B and a small b, so they are heterozygous. Okay, now looking at the colours, we can see that big B gives you brown, because we know big B says brown, and little b gives you blonde, which makes sense. But how is it that this person, who has one copy of both, has got brown hair? And the reason behind that is because big B is dominant, which means, and the definition for dominant now, is that you only need one copy of it in order for it to be expressed. Whereas recessive, you need two copies for it to be expressed. So this person has one big B, and therefore that's the one that's going to be expressed because that's the dominant one. Okay, so here's a question that we're going to use, and we're going to talk about how to do Punnett squares and genetic tree diagrams at the same time. So again, H is a gene for hair length in cats. Big H is long hair, little h is short hair. And the question is, a long-haired male cat was bred with a short-haired female cat. They had 4 kittens, 4 long hair and 4 short hair. What was the genotype of the male? Okay, so let's quickly apply our knowledge for that. Now, so the female had short hair. So she was little h, we know that definitely. The male, on the other hand, had long hair. He could have been big H, or big H, little h. The question is, which one is he? Was he the homozygous one, homozygous dominant, or was he heterozygous? Okay, so they were bred together, and they had 8 kids. 4 were long hair, 4 were short hair. So they had 50-50 split in their kids. So what we can do is have it set up 2 scenarios. So let's suppose that the male was dominant, homozygous dominant. Of course, the female is going to be the same. So what would the children be like? So we can quickly make a Punnett square. So the way we do that is the following. So for the male, it's going to be big H. Female is little h. And if you cross them together, so big H, little h here, big H, little h there, big H, little h, big H. So we can see all of the children are going to have long hair. So 100% long hair. Which is, so therefore, it could not have been this person. Meaning that, or this genotype rather, meaning that the male was actually this one. And we can prove that. So let's quickly do another one. Let's prove that it was that. So we're going to do big H, little h for the male, and little h for the female. So we can see that 50% are going to have long hair, and 50% are going to have short hair, which fits exactly the pattern in the question. So therefore, the answer is that the male's genotype was big H, little h. Okay. The next one. A purple-furred male rabbit was bred with a white-furred female rabbit. Devise an experiment to find out the genotype of the male. So big R is purple, little r is white. So we want to find out what the male was and what experiment could we do. Okay, let's have a look. So this time they haven't told us what the children look like. So I'm going to rub this out. So we know the male was purple. So again, he could have been big R, or big R, little r. And the female had white hair. So female is definitely going to be little r. So what experiment could we do to find out what the male was? Which one he was? So the first experiment we could do is we could, of course, breed them together and see what kind of children they will produce. So let's say that the male was big R. So if I was to breed them together, big R, little r. I can see that all of the children are going to be heterozygous, and therefore they're all going to be purple. If the male was big R, little r, and I was to breed that with the female, big R, little r, and then we can see that there's going to be 50% heterozygous, and 50% homozygous recessive. So so half of the kids are going to be purple, the other half are going to be white. And therefore, we can conclude that by doing the experiment, if all the kids are purple, then the male was dominant. If half of them are purple and the other half are white, then the male was heterozygous recessive. Okay, now just as a side note, we've been doing Punnett squares, but we could also do it as a genetic tree diagram. So I'll give you an example of how that would work. So let's say let's breed this with this. So we're going to write, first of all, parents' genotype. Okay, then you're going to write down gametes. So gametes are going to be, if I was to break this down, that will give me R. And if I was to break this down, that will give me little r. Now, just like in maths, when you have two brackets and you want to expand those brackets, what would you do? So you would do this times this. Then this times that. Then this times this. And then this times that. So that's how you do a genetic tree diagram. And then this is known as the offspring's genotype. And then afterwards you can write down, for example, purple, purple, white, white. And that will be the offspring's phenotype. Okay, this is called a genetic tree diagram. Okay, moving on to this. Pedigree diagrams. So this is also known as like a family tree diagram, but basically what this shows you is that they'll give you a family tree. On the right there will be a key. So the key, for example, says male means square, circle is female, and they'll give you some extra information, and then they'll ask you to calculate a particular genotype of a person. For example, here we can see that they want you to find out what is the genotype of the baby. Okay, so the baby, who's the baby's parents? It is Eve and Phil. So these are husband and wife. We can see that Phil is a carrier of cystic fibrosis. We should know that cystic fibrosis, if you're normal, that's big F, big F. If you're suffering, that's little f, little f. But if you're a carrier, that's big F, little f. So Phil, we can say is big F, little f. And Eve is also heterozygous as well. So what would the new baby be? So again, we can quickly do a Punnett square for that. So if I was to do a Punnett square, you can do either Punnett square or genetic tree diagram, doesn't matter which one. It's just that Punnett square is a little bit faster. And then you can see, okay, big F, big F, big F, little f, big F, little f, and finally little f. So we can see that the new baby has a 25% chance, or 1 out of 4, to be a sufferer. And then it has a 3 out of 4 chance to be normal, and 1 out of 4 chance to be completely normal, not even a carrier.